Right. Hi, everybody. We wanted to give you guys just one more minute to join us, but it's 2:31, so we'll get going here. Welcome to the February Forum. I just want to remind everybody that renewal season is upon us, so be sure to renew your membership with the WPA for 2023. We hope to see you at all our future forum events. Before I get into anything, I'd like to thank our two outgoing board members, Karen and Jack. We really appreciate all the hard work you have done for the WPA for the last 2 years. We hope to still see you guys at all our events, and maybe we'll get to work with you on the board again sometime in the future. As always, as we do with these webinars, you're not going to be able to unmute yourselves. So make sure you use that chat feature in the bottom right corner. Type any questions you might have there. Also, Tim was saying that he might do a kind of more hands-on activity in the middle of it. So if you would like to participate in that, he'll have you potentially call out an answer in the chat box. So just keep a handle on that and submit questions throughout the presentation, and we'll get to them when we get to them. All right, so with that, I hope you'll all help me welcome our host today, Tim Whitfeld. Tim has an undergraduate degree from the University of Wales in the United Kingdom in agricultural economics. He completed a master's degree at Eastern Illinois University in biological sciences and a PhD at the University of Minnesota with a focus on tropical ecology in Papua New Guinea. His previous work experience includes environmental education at the Audubon Center in Northwoods in Sandstone, Minnesota, curatorial work at Harvard University Herbaria. I think I'm saying that right, sorry. Field botany and ecology with the Minnesota DNR through the Minnesota Biological Survey Program. Collections manager of the Brown University Herbarium. He has been the collections manager at the Bell Museum Herbarium since 2019. So welcome, Tim, and thanks for joining us today.
Okay, thank you. I'm assuming you can hear me okay?
Yep, you sound great.
And I'll share the screen here. Okay, I'm assuming that you can see that okay?
Yep, looks great.
Excellent. Okay. Well, thank you very much for the intro and for the invitation to talk to you this afternoon. It's great to be here, and I'm really happy to share this information about the Bell Museum Herbarium and the Minnesota Biodiversity Atlas, which is our online database that is available to anyone that has a connection to the internet. And so using this resource, you can access specimens from the herbarium and see the collections that we have here. So my name's Tim, as Rebecca mentioned, and I'm the collections manager of the Bell Museum Herbarium at the University of Minnesota. We're situated on the St. Paul campus, and many of you maybe have already visited the new Bell Museum. And if not, I encourage you to do that often and as soon as possible. I think you'll find it to be a really good experience. And the collections part, I'm part of the collections unit department within the Bell Museum, collections and research. And so the Bell Museum has not only the herbarium plant collections, but also zoological collections, including fishes and birds and mammals and reptiles, amphibians and mollusks. And these are all housed on the St. Paul campus, just a short distance from the main museum building. And we are kind of the research side of the museum. And we work with the public education and exhibit teams as much as possible to incorporate all aspects of the museum in all of the public outreach and programming that the museum does. So with that, I want to start out by just giving you a brief kind of introduction about how I got to this position and situation that I find myself in now. I grew up in the UK, but I first really, I can say I first became interested in plant diversity and field botany when I came to the US to be a wilderness guide in the Boundary Waters canoe area up in northern Minnesota. And so this is a place that I'm sure is familiar to many of you. And for me, it was a really mind-blowing experience to first come up here and experience this tremendous wilderness that we have in the state. And previously, growing up in the UK, I'd always noticed plants, but I didn't have the skills to differentiate one species from another. And it wasn't until I came to the US and specifically in the Boundary Waters, where I was surrounded by this wilderness and nature and by plants, that I felt like I really wanted to know and actually know the names of the plants and understand more about their ecology and how they interact with each other and with their environment. And so this all happened in the Boundary Waters, and I feel like plants, of course, are everywhere. They're around us all, but often people just think of them as kind of a green background to their lives, and they don't necessarily pay much attention to individual species. And I could say that I probably was in that same camp as a younger person. But after spending time in wild places like the Boundary Waters, I became really interested in understanding plant diversity. And so I followed a kind of a winding career path and eventually ended up here at the U of M as the collections manager of the Bell Museum Herbarium. And I still go to the Boundary Waters as much as I possibly can. So when it comes to, hold on, just having a slight technical hitch here with the slides. Okay, so that's a little bit about my botanical journey, and now I'm going to introduce you to the world of herbarium collections. And many of you may know, have a lot of experience working with these kind of collections. Many of you may not have as much experience, so I hope at least it is informative across that spectrum. And the story of herbarium collections begins in the 15th century with Professor Luca Ghini. He was professor of botany at the University of Bologna in Italy, and he was also director of the botanical gardens in Pisa. And in Guinea's time, these botanical gardens, like the one he was in charge of, generally arose as kind of an adjunct to a medical school in order to cultivate medicinal plants. And Guinea developed the concept of the herbarium as an add-on to his botanical garden. And he developed this method of drying and pressing plants to preserve them for study by his students during the winter and the non-growing season. And so he was about the first person that did this. And this is still the method that we use today. And it allowed his students to have a permanent record of medicinal plants and also to make them available year-round for study. And most of the oldest surviving herbarium specimens from the 1500s, which there are still a few around, were made by medical students who then took these references to refer to in their medical practice. Guinea's own specimens that he made don't exist any longer, unfortunately, but specimens from some of his students and some of his colleagues are still known in collections, in herbarium collections in Europe. And so herbarium collections originally were linked to botanical gardens and medical schools rather than natural history museums and universities, which is generally where we see them today. And so effectively, a herbarium is like a library of dried plants that were collected from many locations and many dates. And in the early days, these specimens would have been mounted and bound together in books, just like the picture here. And this is how Guinea did it in his time. But now we normally mount the specimens on individual sheets that are grouped together in folders with other specimens of the same species, and then they're arranged into genera and families for easy references and filed in cabinets like this. And this is how we do it at the Bell Museum. Now, the Bell Museum is not the only herbarium collection in the world. In fact, there are at least 3,500 registered herbarium collections around the world in universities and some still in botanical gardens and museums and different kind of institutions. And combined, these collections include at least 350 million specimens that date back several centuries, at least 400 years in some cases. And so this is an enormous data set that can be used for many, many different things in the study of plants and ecology and botany. Now, some of these institutions on the screen here might be familiar to you, and these are some of the largest collections in the world. And so you can see that some of these are enormous collections. The Natural History Museum in Paris with 9.5 million specimens tops the list of biggest herbarium collections. But here in the US, we have the New York Botanic Gardens with over 7 million, and the Missouri Botanical Gardens with 6.2 million, and the Harvard Herbarium with over 5 million, and so on. And so there are specimens from all over the world in these institutions. And then, of course, there are many other institutions with slightly smaller collections, which are equally important, I think, and represent really useful collections for maybe at a local scale. And they document plant diversity from a smaller scale than some of these bigger collections, which often are global in extent. So I should just quickly here introduce you to what exactly I mean by a herbarium specimen. And so the specimen includes the actual pressed and dried plant, and preferably it has reproductive structures included, so flowers or fruits or cones or the spore-bearing parts of ferns. All of these are important for identification and tend to be much more stable in terms of morphology than leaves and stems, etc., which can vary hugely depending on the environment that they're growing in. And in addition, the specimen is glued onto a sheet, and then also included is a label that tells us where the collection was made. And this label is what makes the specimen a scientific collection rather than just a kind of scrapbook type thing, which maybe we all did as kids when we pressed leaves between the pages of a book and then came back to them years later and had no recollection of when or where we collected them. So the label is key to the scientific collection. And here's a close-up of this particular label from this specimen. And so the label you can see includes the location, in this case, Itasca County, and the coordinates, so the township and range, and a detailed description of the location, plus information on habitat and associated species, and also the date that it was collected and who made the collection. So fundamentally, the scientific collection is a fully documented specimen that includes vegetative and reproductive parts, that includes a label with the taxonomy, when the specimen was collected, where it was collected, and who collected it. And then it also needs to be properly cataloged and preserved, and most importantly, maybe available for study by other people. And so in essence, the collection is a documentation of a particular species growing at a particular location at a particular time. And it's a definitive, verifiable data point in that sense. So that's a little bit of general background about the herbarium collections and the history, brief history of herbarium collections. And now I'd like to focus a bit more specifically on the Bell Museum Herbarium. The Bell Museum began its life in 1872 when the Minnesota legislature enacted a law that was to provide for the care of specimens that were collected by the Geological and Natural History Survey of the state. And so this is when the state's Natural History Museum began, which in 1967 was renamed the Bell Museum in honor of James Ford Bell, who was the president of General Mills Corporation and an early supporter of the museum. And so the museum began in 1872. The herbarium itself began a little bit later in 1875, and it was a repository for plant specimens from the Natural History Survey. And these specimens were originally stored in a building on campus. So again, I'm having some technical issues here with slide. So they were stored originally on a building on the Minneapolis campus of the botany department. And unfortunately, this building, the old main building, was actually destroyed by fire in 1904. But luckily, the specimens at that point had been moved. And so the specimens that existed in that time did not get destroyed. Oh, there's the old main. Now, in 1887, Conway McMillan was hired as the director of the herbarium and also botany instructor and plant collector. And at this point, when McMillan took over, the herbarium began to take shape, and the collections at that point were curated by the botany department in a new facility on the Minneapolis campus, a building that no longer exists because it was demolished in order to expand the medical school. So now McMillan was an avid botanist himself, and I think he also had a wide range of interests. And I found this picture in the archives of Conway McMillan, Professor McMillan, in his shirt sleeves here, enjoying something other than botany and maybe proving that botanists are a mean bunch that should not be messed with if this picture is anything to go by. He packs a real punch, it looks like. Now, McMillan was a prolific collector of plants, and he himself amassed around 20,000 specimens, all of which are here in the herbarium, the Bell Museum. And here's just a few examples of some of his collections from the early 1900s. Many of his collections came from the Minnesota River watershed, which he was fascinated by and considered to be, as a quote, considered this area to be peculiarly central in location in the middle of the state and remarkably interesting. And so he focused a lot of his attention on the Minnesota River watershed, and many of his collections come from that area. Now, McMillan also oversaw the first major acquisition of specimens beyond just those that were collected by researchers and field botanists working on the Natural History Survey of the time. And he started acquiring specimens from outside the state, and it was the purchase of 6,000 specimens, which were collected by a field botanist called John Sandberg from the Rocky Mountains. Now, this was the first major out-of-state acquisition. This set of 6,000 specimens. It cost the university $500, which was a considerable sum in 1888. But it did provide a really solid foundation for further expansion of the herbarium. And these are a couple, three illustrations or pictures of some of John Sandberg's collections from the western part of the US. And Macmillan himself was a really relentless person in terms of his effort to build a first-class institution. And he hired and collaborated with many other notable botanists from the time, including at the university, Otto Rosendahl and Fred Butters here in the middle and the right hand, Otto in the middle with the pipe and Fred Butters on the right reading the book. These two leading botanists of the time worked at the university in the Department of Botany and collected many specimens which still exist today. And also Macmillan hired Josephine Tilden, who was a student of his as an undergraduate and graduate student. And Tilden actually commented that that everyone feared Macmillan, and we all feared the man and cringed at being called by him in class. So he apparently was quite a intimidating person. But despite this, Macmillan was a close mentor of Tilden, and she became a leading algae researcher and the first woman member of the biology faculty at the University of Minnesota. And here she is sitting on the log in the front row on the right-hand side there. And Tilden and Macmillan ended up co-founding the University of Minnesota's Seaside Station, which was on the west coast of Vancouver Island. And this was an active biological station for U of M students from 1901 through till 1907. And so through Macmillan's tenure as herbarium director, he built this amazing cohort of botanists who really expanded and grew the collection. So by 1914, the herbarium was truly global in extent, much beyond the boundaries of the state, and the specimens now numbered over 400,000 collections in the herbarium in a very short amount of time, which is amazing growth rate. And since Macmillan's time, through the years since then, hundreds of researchers, faculty members, students, and botanists from Minnesota and all over the world have continued to contribute to the collection. And just a very small number of notable names that have been connected to the Bell Museum Herbarium include Margaret Oldenburg on the left there, who collected thousands of plants in the Canadian Arctic, and Arthur Cronquist in the middle, who was a student of Otto Rosendahl, and he went on to become one of the most influential botanists of the 20th century and director of the New York Botanical Gardens. And then also Olga Lakala, who became the first woman faculty at UMD, and the herbarium at UMD is now named after her. She specialized in the flora of the northeastern part of the state. Now, we could do a seminar on any one of these people, on their life and their work, and that would be really fascinating, but of course there's not time for that. And there's many, many other people that have contributed through the years, but we can't unfortunately include everybody in this brief list here. So until 1996, the herbarium was part of the Department of Botany, and then in 1996, the ownership was transferred from the academic department to the Bell Museum. And this reflected a change in research focus of the academic department, shifting from specimen-based work to more molecular approaches to plant biology. And so it made sense for the collection to become part of the museum rather than to remain in the academic department. And more recently, the herbarium has collaborated with state agencies, including the Minnesota DNR and the Pollution Control Agency. And it's mostly thanks to their work that we have such a good collection of specimens from the past 30 or 40 years. And particular recognition should go to the Minnesota Biological Survey and their hard work of botanists like Welby Smith, who I'm sure many of you know. And Welby himself has collected nearly 40,000 specimens over his time with the DNR from across all 87 of Minnesota's counties. And also he's, of course, published several books on Minnesota's orchids and sedges and trees and shrubs. And we at the museum see this type of collaboration with state agencies as really fundamental to our land grant mission and also to the continuation of that original 1872 charter from the Minnesota legislature. And now, of course, that brings us up to the present day with the new Bell Museum that opened in 2018 and the celebration last year in 2022 of the Bell's 150th anniversary. So now let me give you a few numbers related to the herbarium collections as they stand today. So I showed you that table a little earlier with some of the largest collections in the world, with the biggest one in Paris numbering 9.5 million specimens. Well, we're one order of magnitude lower than that, with about 950,000 specimens. And with any luck, at some point soon, we'll hit that million mark. But at this point, that's where we are with the number of specimens that we have in the collection. And of that 950,000, the biggest chunk of it is vascular plants, and around 650,000. And at least 200,000 of those are from Minnesota. And some of the oldest specimens we have are actually local collections. And this one of heuchera is perhaps the oldest that I've seen from the state. And if you look at the label here, it's not easy to see, but here we have a specimen of heuchera americana from Grand Portage of the St. Louis River of Lake Superior, collected June the 24th in 1832 from damp, rocky woods. So this, as far as I know, is the oldest specimen that we have in the collection, certainly the oldest from Minnesota. And so we can say that we have 2 centuries worth, almost 2 centuries, of specimens representing plant diversity from across the state over that entire time and from all over the state. But obviously, we have more than just vascular plants. We have 140,000 lichens from all over the state again, and many countries around the world. And we have at least 90,000 fungi specimens. And we have 50,000 or so bryophytes, including mosses, liverworts, and hornworts. And we also have 13,000 algae specimens. Some of you may recognize this freshwater algae that could be found here in the state. And this is actually a specimen of Cara collected in Lake Christina in Douglas County. And right now we're working with the DNR they have a project going at the DNR focused on surveys of lakes for Caraceae, for this unusual group of freshwater algae. So that's a quick run through of the history of the Bell Museum and the herbarium particularly. And so now I'd like to discuss some of the ways that we use all these specimens, or maybe to put it another way, is what is the point of maintaining a collection of 950,000 dead plants and mushrooms? This is a question that often I get asked. And so I have a few responses to that. And so maybe the first thing is to say that fundamentally, these herbarium specimens provide documentation of plant diversity through time and space. And in this case, we can look at an example of the roundleaf sundew and see that it's known from all of these counties that are in light green and also in yellow. In the yellow counties, these are states where this species is considered to be rare, threatened, or endangered. And the light green, it's present, but not in any, doesn't have any listed status. So in order for these counties to be lit up like this, there has to be at least one herbarium specimen from that county as documentation that the species exists in that area. And so these kind of maps are based upon not just people's observations, but actual herbarium specimens that can be checked and verified if necessary. So herbarium specimens are important for documenting species distributions. And this is obvious from this map that the plant grows in the Northeast. It's very common and scattered in some of the southern states, and also known and quite common from the West Coast. Here's another example of one that's not quite as common, a plant that's quite rare, known from only a couple of locations in the northeastern part of Minnesota, county in northwestern Wisconsin, and then the Upper Peninsula of Michigan. In both cases, this one and the common Drosera, the documentation for the species, as I mentioned, comes from herbarium specimens. So it's really easy to verify the location and the identification and to create these maps. So documentation of species distribution is one really important aspect of herbarium collections. And another is as an archive of reference material for describing new species. So these so-called type specimens are like a birth certificate, basically, for a new species. And a type specimen is the basis of the morphological description that is required when any new species is published. So designating a particular specimen as the type is a requirement when publishing a new name, so that other botanists can look at that specimen and understand your concept of the species based on the description that you've given. So this is an example of a type specimen. This one is Sedum rosea variety Leedii, or Leedy's rose root, that was first collected in Minnesota, and the type specimen comes from Minnesota. And here's the original description and publication of this name that was published in Rhodora, the journal Rhodora. That's the Journal of the New England Botanical Club, and it was published in 1947. And so you can see the first section of that upper paragraph is the description of the species in Latin, which was a requirement for new species names until just a few years ago. There had to be a Latin description. And in addition to that, there would be the naming and description of a type specimen. And so in this case, they mentioned that the authors mentioned that the type specimens from northeast facing limestone cliffs on the north branch of the Root River opposite the Herman McDaniel farm, etc., collected July the 12th, 1942 by John Leedy. And the type specimen is in the herbarium of University of Minnesota, Herb Min. And so this designates this particular species as the type specimen. And if we look at the label here on the specimen, you can see the exact same locality description, where we see north and east-facing limestone cliffs, north branch of the Root River, opposite the Herman McDaniel farm, etc. Collected July the 12th, 1942 by J.H. Leedy. And so designating these type specimens is a really important, or archiving these type specimens, I should say, is a really important aspect of herbarium collections. And each new species, every single species of plant has a type specimen associated with it. That's true for animals as well. So not only are they an archive of reference material for new species, the herbarium is also a reference collection to help with the identification of new specimens. So if you were out in the field and you collected a specimen and you were having trouble with the identification, you could compare your specimen to those in the herbarium that have already been identified to make your determination. And so that's another way that these specimens are used. Now, given the temporal aspect of the collection, you can also think of the herbarium as kind of a low-tech time machine. Now, physicists might tell you that time travel is impossible, but when you're in the herbarium, that's not true. We can travel back in time, and we can look at the change of distribution of particular species through time, as if we were winding back time and watching what happens over the course of, say, for example, the introduction of a new species, an invasive species, into North America. And so we can track the change in species distribution through time by looking at herbarium specimens. And as I mentioned, for introduced plants, this can actually be very useful. So this is an example for purple loosestrife. And according to the specimen record, the first collection of this species in North America came from Connecticut in 1822. And here's the dot on the map to show that. And this is actually from a specimen. Now, if we fast forward a few years, the species is beginning to spread. There's now a few collections from across from New York State, as well as the original from Connecticut. By 1900, the species is starting to spread across New England, and it's starting to get established in the Great Lakes region. By 1950, we have the first collection from Minnesota, from the western part of the state, and more specimens from across the Great Lakes region, plus a few popping up on the West Coast, and one in Mexico as well, and also in the southern part of the US. By the 1970s, this species is becoming quite prevalent across New England, especially the Northeast here, all through the Great Lakes and basically effectively across the entire country, scattered across the country. And by today, the present, this is the most up-to-date that I could find. All of these points represent all of the specimens from across the country. And obviously, as you all know, this has become a hugely problematic species. So this aspect of time travel that we can, that the herbarium offers us is a really useful way to track the distribution of species over time. And I think it's particularly useful for these invasive species. Now, herbarium specimens are also documentation of plant species that are now extinct in the wild. And this example of another wetland species, Juncus curvatus, was known from Massachusetts, and the type specimen was collected in Massachusetts. But now, unfortunately, it's believed to be extinct and has not been seen in the wild since 1928. We do still have specimens, and we have one here. And so these are now the only record that this species ever existed. People have tried to go back to this quite precise location here to find this plant, but they've not been able to find it due to development and degradation of the salt marsh that it grew in originally. And you can see here the people that collected this, Merritt Lyndon Furnold, who was a Harvard botanist, and our own University of Minnesota Fred Butters that I mentioned earlier. They collected this plant in 1916. So these are the only records that this species ever existed. And one of the fundamental uses of plant specimens, and all scientific collections, in fact, is to understand the evolutionary tree of life, and also taxonomy and classification as well. And historically, in order to understand evolution, we tended to use, or researchers use, morphological features of flowers and fruits and leaves and other plant parts. And these were measured from herbarium specimens. And you could compare and contrast species with the idea that those that look similar were likely or somewhat likely to be closely related, although that's not always the case, of course. And more recently, researchers can use the same herbarium specimens and extract DNA from these specimens and use those DNA sequences as a source of data to try and understand the tree of life and generate hypotheses about the different relationships between different families, genera, and species. So these are just a few of the many, many ways that herbarium specimens are used today. And this is just a small subset of all of the different things, all the different ways that people use the collections. And of course, many of these were unthinkable to the people who originally collected the specimen. Furnold and Butters and others in the 1920s or early 1900s had no idea that we would be extracting DNA. They didn't even know DNA existed, and let alone that we could collect it from specimens and use it in the ways that we do today. They had no concept of databases and aggregating data in the way that we do today. But all of their collections are useful for these things, and the new collections that we continue to add, we have no idea what they might be used for in 100 years from now, which I think is what makes collecting a really exciting and important work that we do. So now I want to transition from talking about the history of the herbarium and the ways that we use collections to discuss some of the ongoing work in the Bell Museum Herbarium, because it's important to remember that the Bell Museum Herbarium is not just a static snapshot of plant diversity. It does provide this long-term documentation. And so in order for us to remain relevant and important, we need to keep adding new specimens. And it's my fundamental goal as the collections manager to continue the legacy of Conway McMillan and those people that came before me, who were all dedicated to building this really world-class herbarium collection. And so I already mentioned the Minnesota County Biological Survey and their ongoing work across the state. And this is really a vital source of specimens. And it means that we have a really strong collection and representation of local plant diversity from not just from the early days of the herbarium, from the late 1800s, early 1900s, but all the way up to the present day, and including the past few decades. And this is quite unusual. Many other herbarium collections have really good representation from pre-1950, the early 1900s, but much more patchy collections since then as the trend and focus of biology tended away from field work, specimen-based work into more molecular and lab work. And so, but we're lucky at the Bell Museum to have this continual record all the way up to the present day. And I've been very lucky to have participated in a few collecting expeditions with biological survey staff and botanists and ecologists. And our focus in these cases have been documenting plant diversity up in the northern part of the state, back in the boundary waters, and also Voyageurs National Park. And this has generated many hundreds of specimens that are now in the collection. But closer to home, I've also begun working on some botanical inventories in some of the local scientific and natural areas. And the idea here is to provide a species list and a botanical inventory for each site, and each name on that list is associated with a herbarium specimen as documentation and verifiable documentation of the species that I've seen. And so we can use the specimen as this kind of way to fully document the diversity at any given site. And so I've worked at Lost Valley Prairie, which is in Hastings, just north of Hastings. And this provides the DNR with a species list for these sites, and provides us in the museum with new specimens from this particular site. Now, Lost Valley is a really interesting spot. It's about 320 acres in size and has some really nice dry bedrock bluff prairies with some old fields and a few patches of degraded woodland as well. And my collections at this site over 2 years, 2020 and 2021, included 281 species of trees and shrubs and grasses and forbs and ferns and vines and everything that was growing on the site. And every species is validated by a specimen that's in the museum herbarium. And these are just a few examples of some of those collections that I made. And this is a diverse site, not huge, and parts of it are somewhat degraded, but there's still a lot of diversity here with over 65 plant families that are represented. And in this figure, each piece of the pie here is a different family, and the size of the pie reflects the number of species at the Lost Valley Prairie site. So as you might expect in a prairie like this, or mostly grassland, Poaceae, grass family, and Asteraceae are the two most abundant families, the most diverse families. But we also had a lot of species from the Cyperaceae and the Fabaceae and the Rosaceae, as well as all these other families from across the evolutionary tree of life. So there's a really high level of diversity at this site, which made it an interesting place to work. And so after completing work, although I'm sure there's still more things to be found at Lost Valley Prairie, I've now turned my attention to another SNA, the Crystal Spring SNA in Scandia, which is just south, not far from William O'Brien State Park. And this is a different kind of habitat, mostly mesic and dry mesic hardwood forest with some really dramatic ravines on the bluffs above the St. Croix River. And so last summer I worked with a graduate student from the U here, and we collected 250 specimens, at least 250, representing about 220 or 230 species from this one site. And I hope to go back next year to continue this work. So returning once again to Conway McMillan, the first director of the herbarium, it's our goal at the Bell Museum to continue to build and maintain a collection that includes specimens not just from Minnesota, but from all over the world, just as Conway McMillan succeeded in doing during his tenure. And so with this in mind, the herbarium has a history of bringing in specimens from all over the world, and currently at least 150 countries are represented in the collection. And many of these are older specimens from previous work from faculty and students at the university, and also specimens that came in from outside the university. But there are several other ongoing collecting efforts in other countries, one of which I want to highlight briefly since I've been involved with it for quite a few years now. And this is an example that I just want to briefly mention of ongoing work in Papua New Guinea. And this was the location that I did my PhD work. And my first trip to Papua New Guinea was in 2007. And this was the first of 3 trips I took for my PhD, and then I've been back 5 or 6 times since then, every year or two, and most recently in October this past year in 2022. Now, I've done work in Minnesota here locally and also in Papua New Guinea, and it was quite surprising for me to find out that there are species common to the state of Minnesota and the island of New Guinea, which is in the South Pacific, just north of Australia. So these two disparate countries or disparate regions of the world are united by at least those species on this list, both of that grow in both locations. Some of these are somewhat weedy specimens that I think have been spread around the world by people through the years. But some of them I think are naturally occurring in both locations. And it's really hard to understand and get your head around how a species of Carex, for example, that grows quite common in the state here, could have made it all the way to Papua New Guinea. It's common across the northern hemisphere, this species, and it is found in China, Japan, and then a couple of other isolated mountaintops in the Philippines, and then on top of a mountain in Papua New Guinea. And so how it gets there is baffling to me, but these species are actually common to both Minnesota and New Guinea, and there are probably others as well. Now, Papua New Guinea, as I mentioned, is on the eastern half of the island of New Guinea. The western part here is part of Indonesia, and Papua New Guinea is the independent part of the island. And we're just north of Australia here in the South Pacific. And my work takes place in this area, the Wanong Conservation Area, in close to in the northern part of the country, in the lowland rainforest. And recently published paper in the journal Nature has really highlighted the diverse flora of the island of New Guinea, and it is the world's richest island in terms of plant diversity, with over 13,600 species currently known, and many more, at least 100 have been found and described since the publication of this paper in 2020. So clearly there's a lot of exploration to be done. Now, the village of Wanong, where I've been working, lies within an area that's designated as a timber concession, the Middle Ramu Timber Concession. And most of the communities surrounding the village where I work have opted to take timber royalties in exchange for logging rights on their land. But seeing the destruction that was caused by these logging companies, the Wanong community decided that they would prefer to protect their forest for the sake of future generations. And it was under the leadership of Philip Darman on the upper left corner here, who was a community leader, that the village established the Wanong Conservation Area, Conservation and Research Area, that is an area that's outlined here in red that you can see on the map, over 10,000 hectares that encompasses the land that's owned by all of the 11 families within the Wanong community. And in the year 2000, all of these Wanong families signed a conservation deed, and then they invited biologists to come to their forests. And these visiting researchers provide employment and educational opportunities in exchange for access to field sites and to do their field research. And the village then receives conservation and research payments, as well as salary for working with these researchers for protection of their forest. And this money comes in lieu of the timber royalties that they've given up by not allowing the logging company to come and cut the trees down. And at the very center of the conservation area, in this small rectangle here, is the community has established the Wanong 50-hectare forest dynamics plot. And this is part of a network of similar plots that are found throughout the tropical zone and also in some temperate areas as well. And it's run by the Smithsonian Museum, and it's the Forest Global Earth Observatory, the Forest Geo Network. And this is a network of scientists and researchers, research sites that is dedicated to advancing long-term study of the world's forests. And the Wanong plot, you can see on the map here, is the only one in this Australasian region, and it's the only one that is managed and overseen by indigenous landowners. And it lies in the heart of the Wanong Community Conservation Area, and in this red box here, 50 hectares in size. So that's about 123 acres. And the long side of the rectangle is 1 kilometer, so 1,000 meters. And the short side is 500 meters. And associated with the plot is a field station right in this little tiny clearing here. And here's an aerial shot of the station, pretty basic. And this is to get to the station, it's a 6-hour hike from the closest road, depending, that is, on how much rain has fallen. It could be longer than 6 hours if the road is not passable. Now, an alternative, if you have a spare $10,000, is you could fly in by helicopter and land right here in this small helipad clearing. But generally speaking, I've never done that, and I've always taken the 6-hour hike. So the goal of the plot in Papua New Guinea, and with all of the plots in the Forest Geo Network, is to document and monitor and study biodiversity throughout the world's tropical and temperate forests, and also to monitor long-term dynamics of the forest. And by dynamics, I mean documenting how much individual trees grow, so their biomass accumulation, and how many trees die through the years, and how many new saplings or new recruits come into the forest. And before the survey can actually happen, the 50-hectare plot had to be gridded out into 20 by 20 meter quadrats. And that's the unit that we do the survey in. So this involved accurately placing corner posts for each of the 1,250 of these 20 by 20 meter quadrats that are all contained within the 50-hectare plot. So we used one of these surveyors' total stations to do this, and this allows us to be accurate to 1 centimeter or so over 20 meters. And then after the plot was set up, comes the census, which follows the Forest Geo protocols that is common to all of the plots in the network. And this protocol calls for each stem that's greater than 1 centimeter to be tagged and mapped and measured and identified. And then the entire plot is recensused every 5 years, and all of these measurements are taken again. So we can look at the growth and the mortality and the recruitment of trees in the forest. And this standardized method allows us to make comparisons across the continents and compare forest dynamics from one tropical region or temperate region to the next. And within the Wanong plot, the community has finished 2 complete censuses. Starting, the first one was completed in 2010, the second one in 2015, and we're now overdue for another one that we're trying to secure funding so we can start that as soon as possible. And each census has involved tagging, measuring, and identifying over 280,000 stems within the 50 hectares, and that represents about 580 tree species. So those are just trees. We're not looking at any herbaceous plants, trees and bigger shrubs, anything with a DBH of 1 centimeter or above. And this figure, again, shows all of the plant species, the tree species in the plot. And again, each wedge here is a individual species, and the size of the wedge represents the number of individual stems for each species. So in the upper right-hand corner are the common species, and there's at least 7 or more that have at least 5,000 stems in the plot. So these are these with the bigger wedges of the pie here. And then on the right-hand, lower right, we have species, at least 65 species with 1,000 to 5,000 stems in the plot. And then on the left-hand side, there are 206 species with only 100 or between 100 and 1,000 stems, so not so common across the entire plot. And then in this upper right-hand corner, upper left-hand corner, of that 580 or so species, there are 300 that only have between 1 and 100 stems in the plot. So these are really rare trees, and 45 of these species have only 1 stem. And so these forests this is typical for tropical forests, where you have a few species that are common and many species that are very rare. And so you might have only 1 or 2 individuals in the large area like this. And that's why these large plots are useful, because we can incorporate much more diversity than we would if we had a series of smaller, say 1 or 2 or 3 hectare plots that would probably miss most of these really rare species. And since we map all of the trees, we can actually visualize the distribution of each species to within a few inches. And so this helps show some interesting patterns related to individual species. So some species are restricted to certain environmental conditions. So on the upper left here, that map where the black dots represent the location of individual tree stems, Ficus pungens is a species that's of early succession. So it's growing here in forest gaps from tree falls or small landslides that occur in the plot. And then others like Ficus halliana are restricted to certain habitats. In this case, Ficus halliana grows almost exclusively in low valleys in the plot. And then the third species there in the middle, Needham neeman, is basically ubiquitous and common across the entire plot and grows effectively everywhere without any specialization. Now, in addition to all this ecological data, we also are documenting each of the tree species in the plot with herbarium specimens, and that's part of the work that I'm doing. And this is really important, especially for any ecological study, but particularly in New Guinea, where there has not been much botanical activity relative to some other tropical areas over the years. And it's a challenge collecting in the tropics, because we want to find fertile specimens with flowers or fruits, but these are the best for the herbarium specimens. But unlike in a temperate forest, there's no one season where all species flower and fruit. So one has to be there for a long time to catch the fruiting and flowering cycles of all of the species in the plot. And it's also challenging for identification. We have at least 580 species, probably more, but there's no comprehensive field guide to the plants of Papua New Guinea. So identifying these plants is really difficult. And so often the trees are named just a family or just a genus, and we collect the specimens, and then we try, by looking at other herbarium specimens in other collections, we try to make the identification. And I just wanted to highlight the work here of a PhD student here at the University of Minnesota, Zaki Ezedin, and he's a graduate student in plant biology, and his PhD thesis is specifically focused on the trees of this 1 on 50 hectare plot. And he's taken some really nice pictures that will go along with specimens that he's collecting and be used in a book for the trees of the 1 on forest. And he works with the local landowners from the 1 on community as assistants and botanical experts. They know the trees pretty well, and so that they're a really important part of this work. And he's also working with the National Herbarium in Papua New Guinea, where duplicate specimens of all the collections will be deposited. So that's a brief run through of some history of the collection here at the Bell Museum, past and the present, so you get an understanding of how things came to be as they are now and what we're doing at the moment to continue this legacy of collecting and curating specimens from Minnesota and from many other countries around the world. And I just want to pause for a second here and see if there's any questions about this part of the talk. And then I want to give you a quick demo. I'm hoping I can give you a quick demo of the Minnesota Biodiversity Atlas, which is our online database that we use to keep track of all these specimens. Now, there's a slight wrinkle here because this being the first Wednesday of the month, yes, I was worried about that. Our server is down for maintenance. And so I'm not sure if I'm going to actually be able to do this demo. I didn't think about this until this afternoon when suddenly I was not able to access the database. But what I will do is show you some other online resources where you can find collections information, and you can yourself, and I'll give you the website for searching the Biodiversity Atlas, and you can explore that on your own and come to me, email me with any questions that you might have about that and how to use it. It's pretty straightforward, and so I'm hoping that it'll be self-explanatory, but I will show you these other resources as well. But meantime, if you have any questions, I can address those related to what I've talked about so far.
Yeah, we did have a couple come in. I'm trying to order them so they make sense and flow together. So I'll start with this one. At the risk of sounding ignorant, what are the requirements for submitting specimens?
That's a great question. Thank you for that. Um, well, the first thing is that it's a specimen, like I mentioned, that includes flowers and fruits and leaves and um, and also includes a label with all the information that I mentioned. And it's been collected with permission from the landowner or with a permit if it's on state land or public land. And then you can submit those specimens to the herbarium for accession into the collection. And so we are interested in adding more specimens, particularly if you're doing field work that documents, say, plant diversity at a particular site. If you're interested in making those collections as a way to verify your species concepts, then we would be happy to accession those into the collection. Now, one thing, we don't have unlimited space, as you can imagine. So we tend to focus on taking, first of all, the best quality specimens, but also those that are maybe county records or they're unusual plants, or if it's a common species, that it represents a kind of fills in the gap on the map. So maybe there's a big gap in the one part of the state where not many collections of this particular species have been made. That would be a useful addition to the collection. So assuming it's of the right quality with the correct label and represents either a county record or something that has not been collected from the particular part of the state, then we would be happy to accept it into the collection.
Okay, cool. And how many records, either on average or a range, do you usually get in a year? Submitted to you, excuse me.
Last year we accessioned over, I think it was about 2,500 specimens into the collection. And these were from the DNR, from my specimens, from graduate students, and also we were sent collections. It's a bit like trading stamps or baseball cards. We often send specimens, maybe duplicates if we have 2 or 3 from the same species from the same location. We might send a duplicate to University of Wisconsin, and they reciprocate and send us specimens from their collection. So we trade, just like baseball cards, we trade specimens. And so some of that number, 2,500, might have come from other collections outside of not just researchers here at the U or from the DNR.
Okay, one just came in that I think will be a good follow-up to that. Do any of the smaller universities in Minnesota have herbariums?
Yes, they do. UMD has a really nice herbarium. And actually, I will mention this briefly when I'm talking about the Biodiversity Atlas. St. John's, St. Ben's herbarium, St. John's and St. Ben's colleges have a herbarium. Mankato State and Moorhead State and St. Scholastica, St. Cloud, all of these have smaller collections, most of which are somewhat local to the area of the state that they're located in. And so they represent really good example. They're really valuable for that because they often have local collections made by students that we don't have collections of here at the U. And so the small collections often fill in the gaps. And so they're really valuable in that sense.
So it sounds like you guys collaborate between.
Yes, we do. Yeah.
Okay, cool. Here's a slightly longer question. Do you guys have any trouble representing more abundant or common species? Because we assume there's a proclivity to collect samples that are more rare.
Yeah, that's an interesting and really important question. Thank you. There is no question that weirdly, often common species are underrepresented in the collection. Dandelion is a good example. We know that that occurs almost everywhere, but we don't have specimens from across every county in the state. And so people do tend to overlook the commonplace species, thinking that there's no point in making a collection. And they focus on unusual, weird plants that are more interesting, perhaps. But it is important to have a representation of everything. And that's why, for me, doing these inventories at local scientific and natural areas is important, because I'm collecting every and so we get this representation. And there's been interesting research done on the biases in collecting. Common plants is one. Strangely, it's a bias against the common plants, as you mentioned, but also there tends to be a bias towards collecting in easily accessible areas, so along roads or trails. Many people often are not that interested in bushwhacking for miles to get to out of the way places. So you tend to get these patterns where collections come along roadsides, and people tend to focus on wildflowers rather than grasses and sedges, things that are much more difficult to identify. So in many cases, those difficult groups are underrepresented in herbarium collections, which is another reason these general collecting efforts where you get everything, irrespective of its rare, how rare it is or how pretty it is. These collecting efforts, I think, are really valuable to try to avoid that bias.
I don't know how you're going to answer this one, but I had this question myself. Your mushroom slide kind of threw me. Like, how do you guys show mushrooms, store mushrooms? Do you have slices of mushrooms? You just dry them out in a room? I don't know a lot about mushrooms.
Yeah. No, I'm certainly not a mycologist, and I don't collect mushrooms. But we do not. What I do know is that we don't press mushrooms like we do for vascular plants. They are simply dried in a plant dryer, whole, 3-dimension. So they shrink a little bit, but they maintain their general shape. They often lose their color, but not always. Some of them are still brightly colored. And they then get stored, instead of being glued onto a sheet of paper, we put them into little boxes. In fact, I don't actually have, oh no, just a second, excuse me. We put them into little boxes like this, depending on the size of the collection, of course. This one is a small specimen from up in the boundary waters, and it's wrapped in tissue paper, you might see, inside the box. And inside the box is other information related to the collection. And then we will glue the label. Eventually, when I've made the label, it gets glued onto the outside of the box. And then these small boxes get stored in bigger boxes. So it's like a Russian doll type thing. And if it's a huge mushroom, it has a huge box, and this is a slightly bigger box here. You can see different sizes. So we have all different shapes and sizes that then have to get fitted into the bigger box, just like a big game of Tetris. And those boxes get stored in the herbarium cabinets and cataloged and imaged just like the regular specimens.
Okay, that makes sense.
Same thing with lichens. You don't press a lichen. That gets collected on the substrate that it's growing. So it might be collected with a chunk of tree bark, or the researcher might break off a piece of rock if it's growing on the rock, and that gets put into a small packet which is glued onto the herbarium sheet. And mosses also just get dried and not pressed. But algae, depending on the species, may be pressed just like a regular plant. So seaweed, for example, leafy algae will be pressed and just like a normal plant specimen.
I suppose that makes sense when you say it that way. Let's see here. A couple came in while you were talking. How do we, as field scientists or collectors, ensure that we are not threatening the survival of a rare species?
That is a really good point. And so the rule of thumb is that you would never collect if you've only seen, let's say, a dozen or a couple of dozen individuals of that particular species at the site that you're working in. So you wouldn't want to collect that one that you've seen, the only one. You really wouldn't want to be the person to extirpate a species from a particular area. And so we have to be very careful about this. And the permits that I have from the DNR, for example, have it very well clearly laid out. Only can collect a single specimen. If there are 10 or more individuals, you can collect one specimen. And even then, you wouldn't dig up the roots. You would just snip the top off it. And so we have very careful protocols to avoid that problem. There are terrible stories of collecting a particular species from on top of, I think it was Mount Washington in New Hampshire. It was a species of potentilla, I think, and something closely related to potentilla. And it was known to really only occur on Mount Washington. And botanists flocked to this site to collect lots of interesting plants, but this one in particular, and they effectively wiped out the population. And so it's a bad story and a good example of needing to be careful about collecting, over collecting, and particularly for rare species. So one approach is to, rather than collecting an actual specimen, is to take a photograph, digital image, which could be added to a database as documentation that the plant was in that location.
Sure. It looks like we have just over 20 minutes. Do you want to try the resources? We can come back to questions after.
Yes, okay. So I will mention, it's such a shame, and I apologize for this, but there's nothing we can do about it. So the Biodiversity Atlas is the Bell Museum's database, online database that includes records from not just the herbarium, but also the other natural history collections, so the animals, the birds and mammals and fishes, etc. All of them combined into this one atlas or one resource that can be searched by anyone that has access to the internet. And so it's kind of an extension of the collection in that way, that we want the collection to be much more accessible beyond just having to come and visit the herbarium. You can now look at the collection online because we have high resolution digital images. All of those pictures of specimens I've shown you came from the Biodiversity Atlas, and records that can be downloaded as an Excel spreadsheet. They can be mapped, the data points can be put on a map and visualized in that way. And so it's a really useful resource for tracking biodiversity specimens that we have at the museum. We don't have everything online at this point from the herbarium, but we're gradually working towards that goal. And right now we have about 250,000 records and images of plants. Every specimen from Minnesota is represented in the atlas. There isn't necessarily a photograph of it, but there's a record. The record is in the atlas. And so you can be pretty certain that if you're searching for Minnesota species, you're getting a really good representation. And it's not only specimens from the Bell Museum in the atlas. In fact, so actually one thing, you can see the web address there if you are interested in checking out what we have in the atlas. And I only wish I could give the demo, but that's just not going to happen, I don't think. So just a brief background on the atlas. It's funded through the Environment and Natural Resources Trust Fund, administered by the Legislative Commission on Minnesota Commission on Minnesota Resources. And the first phase of it was funded in 2015, and that was to incorporate all of the Bell Museum's records of natural history collections just from the museum. But we then expanded in 2018 with a second round of funding, and that was adding hundreds of thousands more records from some of these outstate university, college, smaller herbariums that I mentioned, as well as state agencies like the DNR and the Pollution Control Agency, and the Science Museum as well. And not only actual specimen records, but for example, the Minnesota DNR provided us with all of the expert observations from their vegetative plots that they've done across the entire state for decades now. This is hundreds of thousands of records of species from their vegetation plots identified by expert botanists, so we can be pretty confident in the identification. There's also wetland plants from NPCA that might be useful for your work, and these are all represented in the atlas as well. And then this year, we have been recommended for funding for a third phase of the atlas. Now, this is, of course, pending legislative approval, but we are in the list that is part of the bill that would need to be voted on by the full legislature sometime this spring. And this expands the atlas to over 2.5 million records, and we bring in more expert observations and specimens from different additional universities, Morehead State, St. Scholastica, also records of observations, more observation records from the DNR, the Minnesota Ornithological Union, U.S. Department of Agriculture, as well as insect collections from the U here and also the UMD insect collection. So we're expanding. Right now, there are no insect collections, or very few, but we're going to be remedying that in this third phase of the atlas. And the big part of the funding that we get goes towards students who digitize herbarium specimens. So that means they take a photograph and then they transcribe the label data. So everything is combined into the atlas and available for anyone to search. So there's what I was going to do, but unfortunately I cannot at this point. But what I can do is mention that the atlas is built on a database architecture called Symbiota. And this is an open source biodiversity management software that's funded through the National Science Foundation. Project is based at Arizona State University. And there's a network of these Symbiota-based portals, they're called 54 of them, in fact, including the Minnesota Biodiversity Atlas. And these incorporate data from preserved plants and animals and fossils and fungi from many different collections around the country. So I just want to show you that list here. I just optimistically, no, it's still not working. So this is the list of the current Symbiota database portals that exist, and they include things like Arctic lichens and California herbarium specimens. There's a consortium of all of the Midwestern herbaria, including ours. Our data is also on this Midwest herbarium portal. There's bryophytes and lichens, the consortium of Northeastern herbaria across New England and New York and Pennsylvania, New Jersey as well, I think. And you can see there's a wide range of different portals that are all searchable. And one of them that's interesting is this SciNet. And this one I could perhaps briefly show how it works, because it's quite similar to our portal. SciNet is a regional network of herbarium collections across the state, including Arizona, New Mexico, Midwestern, and it pulls in information from all of these different portals and kind of aggregates it. So the Midwestern Herbarium portal, for example, is this is the main search page, and it includes collections from all of these different universities and herbarium collections around the Midwest, including the University of Minnesota, Michigan, Illinois, etc. All of their data is aggregated into this one search engine that can be used to look for specimens from across all of the different collections. So, for example, if we look for a common species like red maple, we can see what pops up. And we can see there are 16,000 records of red maple that are documented in this consortium. And this is the way the Minnesota Biodiversity Atlas works. It's built on the same architecture, and you search for it in the same way. You can map the specimens. You can download a spreadsheet that includes all of the data that can be imported for use in other programs, software, etc., for analysis and whatever use you might want to make of it. So these are really useful aggregator websites that pull together these different existing Symbiota portals that allow you to search across multiple universities. And in the case of the Biodiversity Atlas, you can search for Bell Museum specimens, you can search for UMD specimens and Minnesota DNR vegetation survey. All of them are included in the atlas, and you can search everything at once or just one individual collection, whatever fits your needs. And so these kind of small scale aggregators work at a regional level. Some of you may be familiar with the bigger scale aggregator, such as the iDigBio, which is the integrated digitized biocollections website. Now this is pulling in information from specimen records from many of these different Symbiota portals and other. Allows you to search. Right now they have 137 million records, natural history, all based on specimens. And so one can search for anything on this. So we could look for example, Drosera rotundifolia, the live demo is always a high stakes, high risk strategy. So here, and it's actually working, which is miraculous. Here are the records in the iDigBio database of Drosera rotundifolia, the round leaf sundew. And all of these Minnesota specimens would be probably from Bell Museum, or many of them would be. This one, MIN, this is Bell Museum collection. And you can then click on the view the record and look at the specimen. This one is from Polk County in west central Minnesota. So these aggregators, these pull data from all of the different online databases that exist. What we do is publish a snapshot of our data that is then harvested by iDigBio and incorporated into their database. And periodically we publish a new snapshot and they harvest that. So they get the most up-to-date records that we have in our collection. And then one final aggregator that I want to mention is the GBIF, Global Biodiversity Information Facility. And this is another aggregator that includes specimen data, but also expert observations and species lists. And so this one is even bigger. This is 2.2 billion records and from all over the world. And this one is, again, you can search for anything that you're interested in. So if we go back to that Carex echinata that I mentioned as being common across the world to Minnesota, and also one of those examples that's known in Minnesota and also in New Guinea, and here's evidence of that. These are specimen records or expert observations showing you the complete distribution of this species around the world. And so these data sets can be downloaded, both the iDigBio and GBIF, as spreadsheets for whatever purpose you're interested in. And I think they're really remarkable. And this is what I was mentioning earlier, the way these specimens are now being used in ways that the people that collected them 100 years ago had no concept this could be possible. And this is really fueling a resurgence in interest in collections from not just botanists, but also ecologists who are looking at all kinds of questions. And one example might be plant distribution and how this species came to be in these disparate locations, the biogeography of species. It's much easier to do that now when we have these really thorough representations of the range of the plant. And so that's just one example. You could also imagine all kinds of different ecological studies that could be done with these data sets. And after a period of when specimen-based research had been considered to be somewhat old-fashioned and out of date, ecologists are now discovering this great resource of 350 million plant specimens. And now these are being aggregated in a way that makes it much more useful for people to use the collections. And so I think I'll wrap up there. Again, apologies that I couldn't do the demo that I really wanted to, but as I mentioned, if you have any interest in more information, then just let me know, and I can send you some more information about the atlas and how you would access it and how you would use it. But I think you'll find it's quite straightforward and should be somewhat self-explanatory. And so just brief acknowledgement of funding for the work that we do, for work here in Minnesota and Papua New Guinea, and support for the Atlas from the Environment and Natural Resources Trust Fund, support from the DNR, and for that work in New Guinea, the community of Wanang is, I'd like to thank them as well. And thank you all. And as I said, if you have any more questions, now we could, I don't know if there's time to think about those now.
Yeah, we have a couple minutes if you're willing to keep going for a little bit.
Of course, yeah.
All right, let's see. Okay, does the database include updates in nomenclature, and is there any lag that we should be aware of or anything like that?
Yeah, we try to keep up to date with the nomenclature, and that is a difficult moving target because constantly there's updates. we focus for North American plants. We use the flora of North America as our standard, and as the volumes of that publication come out, we try to keep up with them and update the nomenclature. But there is a lag, and so the way we set it up though is that we connect old names and new names as synonyms, and so one can search for an old name, and everything that's filed under the new name will come up, both the old name and the new name. So we have these things linked together if it's done properly. And so you should find that the name, you could use an old name or you could use a new name, and you would get specimen records for both things that are filed under both names. But fundamentally, we do try to keep up with the taxonomy, and we update names accordingly, but there will be a lag. So it's not an instantaneous thing.
So if you can't find something, maybe try the old name.
Yes, yes.
Sure, sounds good. What are your thoughts of iNaturalist?
I think iNaturalist is a really terrific resource. I think it's especially useful for documenting invasive species, those that are really common and easy to identify. And I think that for any species, it's really useful because there's enough people looking at it, helping with identification, that it can be really useful. If you're using the data from iNaturalist as a resource for documenting plant distribution, you would need to be careful and curate that data set carefully to make sure that every record there is really what it purports to be, because there are, of course, errors. But overall, I think it's a great way to get people engaged and interested. And it's easier for people to take photographs and post them online than it is for people to collect specimens and deal with that hassle. And so this is a great way to document species and have the community kind of a collaborative effort to curate the data set and make it as the best quality it can be. So overall, I think it's good. I think it's really a great way to get students involved. I know that some classes here at the U, some of them will collect specimens, but they also have students make records in iNaturalist as a way to document species diversity, and students get into that in a big way. So if it generates excitement like that, then I think it's a good idea.
Sure. All right, the last one that came in, I don't know, there's some pushback from some of the other people in here, but Sean was wondering what vascular plants believed to be extirpated in Minnesota do botanists and enthusiasts search for? Some people are saying that's a secret and you can't answer that, so.
Well, I'm trying to think if I know of any that are believed to have been extirpated. There are some. One example that's quite interesting is there's two species, or three, I guess, two species and wild rice. There's Zizania aquatica, Zizania palustris, and the Zizania palustris has two varietal names as well. Zizania aquatica, we have one specimen of that, true Zizania aquatica from Houston County from 1928 or something, an old specimen, and has not been found since that time. How it got to be there is not clear, and whether it's still there is not clear. Whether it's been extirpated or whether it was never common and the population came and went very quickly is hard to say. But it's an example of a species that we have a specimen from. It's documented. It is true Zizania aquatica, unlike and all the other specimens we have are Zizania palustris, but it's not been found since that time. And so it's an interesting case of a plant that might have been extirpated, and people are still looking for it. And there are sure to be others that I'm not remembering off the top of my head.
For sure. I think that's all the questions that have rolled through, and that's good timing because we're just about at 4 o'clock. So we really appreciate you coming in. That was really interesting. I really like that slide where you showed the Minnesota and the Papua New Guinea stuff. That was really interesting to me. So
yeah, it's mind-blowing to me.
Yeah, exactly. I was like, how is that possible? So
how
that's really
possible?
cool. Yeah, exactly. So thank you so much for joining us today.
Well, thank you, and great to meet you.
Yeah.
Spend time with the group.
Thanks everybody for joining us.